Welcome to our Ask the Expert series. Today, we're talking about safety and performance measures and acceptance criteria. We have two speakers today. The first is Laurie Mitchell, founder and president of Criterion Edge, a global medical and regulatory writing firm serving the medical device, pharmaceutical, and biotech industries. Laurie has over 20 years of experience in medical writing, safety and pharmacovigilance management, and regulatory reporting. Having provided regulatory and medical writing solutions to many pharma and medical device companies, both large and small, she is a proven leader in designing practical strategies to meet current global regulatory challenges. Laurie is a published author and holds a Master of Nursing from UCLA. Our second speaker today is Dr. Sarah Chavez, a principal medical writer with Criterion Edge. Sarah has a BS in biochemistry and a PhD in molecular and cellular biology with 25 years of laboratory bench experience and has taught at the university level for the past 15 years. She has extensive scientific writing experience in an academic environment, as well as for several large textbook publishers. Sarah's strong background in basic research and her extensive experience with regulatory writing will allow her to assist companies with IVDR readiness as part of the Criterion Edge team. Now I will welcome Laurie to give an overview of today's session.
Hi everyone, thanks for joining. Today is a live Q&A session. We've got it scheduled for 45 minutes. And what we're going to be talking about today very specifically is safety and performance endpoints, objectives, appropriate, clinically relevant, and acceptance criteria. We get a lot of questions about this, sent to us, or when we engage with some of our clients, there seems to be a lot of questions around safety and performance and how you set those endpoints and objectives, and then how do you determine and identify acceptance criteria. So I think we're going to have a good interactive session today. Thank you very much, and we can get started.
This is our first question today. It's actually a two-part. How do you define acceptance criteria, and how do you define safety and performance measures?
Yes, so this is a really good place to get started, just to make sure that we have all of our definitions consistent so that we're using the same terms that you think we're referring to. So when we talk about safety and performance endpoints or measures, these are going to be the types of data pieces that must be appropriate and clinically relevant in the context of what can be considered state of the art in the field. So ultimately what that means is that you want to identify ways in which across the field this particular type of test or this particular type of technology is measured or defined. How do we define success? What are the certain types of performance measures that are commonly used? So you can often get this type of information from clinical practice guidelines or the CLSI guidelines. So these can be, there are going to be certain acceptance or performance measures that are going to be fairly consistent across all IVDs, and then some that may be specific to your particular test. Now, in contrast, an acceptance criteria, this is derived from the data that you've collected. So within your state-of-the-art section, you're going to be collecting extensive data, hopefully, related to performance and safety criteria. The acceptance criteria is then, that's going to help you determine the acceptability of your benefit risk ratio by being the data that's actually collected, the range that you've identified based on your analysis and your collection of safety and performance measures. So the criteria are the data, the safety and the performance measures are the objectives that you're looking for as part of your state-of-the-art search.
Sarah, if you could clarify a little bit further, the acceptance criteria come from the competitive landscape. Isn't that correct?
Yes. So within your state-of-the-art section, I know we've done several webinars really going into a significant amount of detail on the state-of-the-art. A big part of that is setting your competitive landscape. So you need to identify competitors who use very similar technologies and approaches who you would define as your most close competitor. By identifying the data set from those competitors and collecting safety and performance objectives from those competitor analyses, that's ultimately how you're going to set the landscape. It's kind of like saying, if you want to define what's the acceptable range of performance for your particular device, you find sort of who's your closest in the field. Who are the direct competitors who have a similar test? How do they perform? If their performance range is, their sensitivity rates are anywhere from 95 to 99%, that range is your acceptance criteria. Their 95 to 99% represent the numbers that are reported in published studies on your competitor devices.
Very good, thank you.
All right, let's go ahead and get to our next question here. How different is the process of identifying the acceptance criteria compared to medical devices?
Really, it's not going to be that much different than what we're used to doing for medical devices. You still are expected to meet the parameters of safety and performance to be able to be considered as being acceptable for use. So you still must show that you meet your intended purpose as stated by the manufacturer, and that there's an acceptable level of side effects, which is the safety part. So that's kind of the two parts of it. The safety is really showing that there's an acceptable risk, whereas the performance is telling you basically that you've met your intended purpose as stated within your purpose statement. So it's all going to need to sort of come together here, but really when we're looking at it from IVDR terms, it's the same process that we're used to doing for the MDR.
I would like to add, and there may be additional questions coming in about this, but what I would like to add is, how do you derive the competitor data set that you're looking now for acceptance criteria to pull their results essentially on a safety measure or a performance measure that you have identified for your device that your competitors also report on in the published literature. So how do you find that published literature? How do you define this? In our experience in the medical device world, which I feel certain enough to say that is going to be repeated in the IVD world for IVDR, is that you need to run a competitor device search, literature search, systematic literature review, where you're screening those returned articles and you're establishing the included articles as part of your search and review process. And then from those included, selected articles that you have methodologically searched, screened, and selected according to criteria, it's from those articles that you're pulling essentially data that is going to begin to build the acceptance criteria ranges for each one of your safety and performance objectives. So the caveat that I want to get across there is that this is must be a methodologically robust search of your competitor device and screening and selection. Because as I say in many of my webinars and other things, it's data. And when it's data that you're looking at in the published literature, the notified bodies are very, very specific about how you search for them, how you screen them, what you exclude and what you include. Because at the end of the day, we're talking about data. So I wanted to throw that out there as a point to be made for systematic literature review methodology to establish these acceptance criteria, to support the establishment of the acceptance criteria.
Yes, that step is definitely one of the more time-consuming steps, but it's also one of the most fundamental to the building out of not only your PER, but also has to feed back into your PEP, because it all links back to your intended purpose and use statements. You need to know what you're detecting and measuring. You need to know what its function is, and you need to make sure that any of the studies regarding safety and performance tie back to that defined intended purpose.
Great.
All right, before we get to our next question, I just want to remind all of our attendees to send in your questions using the Q&A button on your Zoom menu bar. And let's get to our next question here. What sort of safety and performance related claims can we make? Is it similar to marketing claims? Also, how do we justify them?
Yes, this is a really common question that we get quite a bit from clients who are new to this whole idea of writing a PER. Really, we do actually consult with the marketing groups. We do need to know, they're usually very helpful in understanding the landscape in terms of who your competitors are from a marketing perspective. But it's important to remember that their purpose is very different. Their purpose is to sell the product, not necessarily to have provided all of the necessary data and the process by which you're measuring that data. So it's important to sort of be able to distinguish between what's a marketing claim and what's an actual safety and performance measure in your intended purpose statement. So you're going to want to make sure that your IFU and your labeling and your intended purpose statement are aligned with the data that you have identified as part of your PER readiness. You would want to make sure that you're clear on who your intended population are, what the indications are, the specific safety and performance measures. So it's all very much data-driven in terms of the safety and performance claims, but you can certainly bring in some of the marketing individuals early in the process to help you start to identify those competitors and to at least get you off running in the right direction. You just want to make sure that the data is what's driving your reports of safety and performance measures, though.
You know, Sarah, I'd like to add too that I just saw come up in our chat box that, you know, a call for potentially registering for our February 23rd webinar on IVDR Essentials Performance Evaluation Report, Early Planning and Preparation. And this question goes right to the heart of that. What is that? What do we need to do early and put processes in place that will then move through the other departments, into other departments and other areas. So that early planning and preparation then will inform downstream decisions from regulatory, from clinical, from the technical and analytical side. And one of the key questions to begin to ask yourself early on is, what are our safety and performance objectives that we're looking for in this device, analyte, whatever your subject device is? What are the key components of that? If you begin to define it, because I know you're doing this anyway, if you're developing a new analyte or you're developing something, your design team is talking about, we want it to do X. And we want it to do it by this kind of technology, but we want it at the end of the day to be able to do X, Y, and Z. And so that is the birthplace of your safety and performance objectives. Is it what is it that you want it to do every time? And what are its competitors that exist out there in the marketplace? What are they doing? And are they measuring the same performance, the same safety measures that your new device is intended to do? And do they have the same intended use, the same intended population? So this early planning, safety and performance objectives always come up at the beginning, because then it flows through the rest of your organization, all the way out to your PMPF planning activities. If you're going to collect data proactively in the post-market setting, why not collect data that speaks directly to your safety and performance objectives, rather than obscure data that you really can't use in the PER process? That's what this is one example of that. I wanted to throw that out there for your consideration.
And for instance, if you're brand new to the process, by bringing in the marketing folks, I usually have them kind of just describe their device or their test to me in marketing language. So as they're explaining it to me, like for instance, one client may say something like, our test is known to, it doesn't require this added step, so it's faster. It's the fastest one on the market. That's great. That's a marketing claim that my test takes an hour and most tests could take days or could take 2 hours. That's a marketing claim. What you have to do when you are starting to think of this in terms of a performance-related claim, you want to show how does that performance, how does getting the results in an hour as opposed to days, how does that directly relate to clinical performance and potentially safety indications downstream? Is this a very time-dependent diagnosis? Is this something that is measuring the device performance in real time? If this is a glucose measuring test and you're trying to identify whether or not somebody's going into DKA or some sort of is having a high glucose or a low glucose, time is very important for their clinical, the clinical benefits of that device. If it's something more long-term, that might not be as strong of a performance measure, but it still might be something that could be relevant. So the marketing claims really do give you kind of a direction to start with, but you have to be able to justify why that particular claim is directly related to safety and performance.
Nice, thank you, Sarah. Let's get to our next question here. Acceptance criteria seems subjective amongst auditors. What do you consider to be the objective criteria to measure safety and performance?
So really the acceptance criteria are pretty well defined. Your acceptance criteria is going to be the range of data that you've identified. So again, taking that the glucose testing example, if you have a particular test that's able to identify high glucose levels with the sensitivity of 89%, and then you have a newer test that's now one of your direct competitors, and that bumps that range up into the 90s. You would want to make sure that you are collecting your acceptance criteria from your competitors, not necessarily all of the alternatives. There may be a way of testing for glucose that uses a different methodology or a different type of technology that is less sensitive. If you were to take that low, the 89% as part of your acceptance criteria, you're kind of setting the bar low for yourself. So I think that's kind of what the question's asking in terms of this, how subjective it is. It kind of comes down to who do you define as your competitors? If you appear to have selected competitors who are basically anyone who has a test that detects glucose levels, that's not necessarily truly your competitive landscape, because you may be identifying technologies that are much older, are far less sensitive compared to what your type of technology should be. So in that respect, it really does come down to that planning stage, clearly identifying who is a competitive test and what is an alternative diagnostic methodology, and making sure that you are collecting from the right types of data as you're developing these acceptance criteria. But really the objectives is to define your competitive landscape clearly, to identify relevant safety and performance measures, and then to identify that range of what's considered to be an acceptable range of each of those different safety and performance measures based on your closest competitors.
Now, Sarah, could you expand a little bit further on, remember, we're establishing a range from your 5 competitors, let's just say, and their published literature. And so a range is a range for safety objective number 1. The range is 1 minute to 5 minutes, whatever that range is. So that's a range. How is that range then translate into the ultimately into the analysis of your own data, the subject device data, against that range? Does it need to fall within that range, your subject device's own data, or does the range change if it's a safety or a performance objective? And I think you know what I'm asking. How do you choose? How do you choose the bar?
So if you think about this is providing sort of that like the sweet spot, the range in which you would like your device to fall. But really you're looking at sort of like the high end and the low end of these ranges. So if you're talking about a safety objective, and let's say that there is a risk of a delayed test result or something, or a false negative, let's say, that directly informs safety. So if the rates of false negatives are anywhere from 1% to 5%, you would say, I should be at least lower than that high end of the range. I should be lower than 5% to be considered state of the art. So if my competitors are in that 1 to 5 range, I would like to be below that top end of that range. And it's kind of the opposite for a performance measure. If your sensitivity is 95 to 99%, you would want to be at least that 95%, hopefully above that in the range to be considered the state of the art. So you're giving yourself these ranges, but really what you're going to then do in your APR and your CPR is to show the data for your device. And you're going to say, based on what I've established as my acceptance criteria, derived from the data from my closest competitors, I need to be at least this good or like this good or either above or below that measure.
So again, to repeat, when you're identifying the acceptance criteria ranges, which we commonly do as part of the SVR and the state of the art, because the competitive landscape is a part of the state of the art conversation. And then you culminate that in like, we've looked at our competitors and they are these devices. And we did a competitor device search on these devices over 5 years. And we looked at all of those results and included all of the results that met the articles that met our inclusion criteria. And one by one, we went through each article and we extracted the performance, the data for each performance and safety objective if it was reported in the article. So you're going to have a table that has a lot of rows, which are the competitor device search included articles. And then the columns are going to be the safety and performance objectives named. And then for, Smith et al. 2021, for this performance objective, it's going to be 1 minute. And this safety objective, it's going to be whatever. And then you're building that range. That's what that table looks like. And at the bottom, the range for the performance objective number 1 is 1 minute to 5 minutes. So that's a performance objective. And so just as Sarah said, so you have to translate that now over to when you're now assessing your own data, your own subject device data. That's the whole point of this. And that's done in the PER or the CPR for that matter. And then, so for safety, you need to be no higher than the top range for any safety objective. Your device should perform at least as well or better than the top number from your competitors. Whereas for performance, it's the opposite, as Sarah said. You should be at least as good as the least performing of your competitors. So that's the bottom range. We just wanted to make that very clear. That's what has been acceptable out in the clinical evaluation in the MDR world. And we have no reason to think that it wouldn't be the same for IVD.
Mm-hmm. All right, let's get to our next question. What are the biggest pitfalls or Achilles heels to watch out for in identifying safety and performance measures?
I think at least in our experience so far with the IVDR world, they're kind of the same things that we experienced in the MDR world. It's trying to make sure that you have something that is reported regularly and reported consistently among your competitors. And this goes back to making sure that your plans are in place to be collecting this data proactively moving forward. If you have a particular, let's say that you have a particular performance measure and you use one scale to measure it, but literally everyone else in your field uses a different scale by which they measure success or performance. That's going to make it really challenging for you to directly compare your device to your competitor devices. You may need to reevaluate your own internal data for your device to make sure it's in alignment with your competitors. There could also be some just general inconsistencies in whether or not these measures are actually reported. So you may just find that your particular test, there's just not a whole heck of a lot of data out there on your test. So in that case, we usually cast a very wide net in terms of trying to extract the data for any safety and performance after we've identified those competitor articles. We basically will go in and try to extract any type of measure by which these competitors are measuring themselves, and then can use that to help us inform whether or not we have what seems to be the most commonly reported measures of success. You can also get some really good insights from clinical practice guidelines. That's usually a really good place to start if you are just not entirely sure. But ultimately, it's going to come down to the data sets that you've pulled. If the clinical practice guidelines all report a particular measure, and yet the published data does not report that measure at all, you may need to identify additional safety and performance measures that will allow you to make that direct comparison later on.
Yeah, that's an excellent answer. And I would add in the caveat is that more is not necessarily better. Some of our clients want to make several safety and several performance objectives, and as in 3, 4, 5 each. And that the notified bodies are in the MDR world, they're not looking for that. The more is not better. Relevant is better. Now, you don't want to choose one safety and one performance only, but sometimes safety is really the rates of all AEs. That's not very common, but sometimes we do have to revert to that. But I would say typically there's 2 or 3 at the most of each acceptance criteria. So choose wisely, choose where you have the best data, you know, the most access to competitive data. And that it relates to your device. But you do not need to be all-inclusive, nor do you need to choose every single competitor that may or may not be very relevant to your device when you do your competitor device search. We commonly just ask for the top. What are your top competitors? And not everyone that may exist out there in the world, but the ones that are the ones that do the most sales, that have they do a lot of publishing on their device, and so you have good access to data.
Okay, here's our next question. How do you determine the sample size for design verification and validation?
Really, this is going to kind of come down to, and assuming the sample size and the design is referring to the competitive landscape itself, sort of how do you define your competitive landscape? How do you collect data to determine your acceptance criteria? It's going to be based on a well-designed search strategy. So you want to make sure that you are capturing the correct amount of data from the literature. If you have a particular test that has a very extensive publication record, you would want to certainly limit yourself to maybe the past 5 years, and then make sure that let's say you have studies, you have hundreds of studies potentially. That's the point at which you may want to limit yourself to some of the meta-analyses or only randomized control trials of patients or a certain number of samples. You may want to say only studies that include more than 100 patients. something to that effect. You want to make sure that ultimately the decisions you make during that, that search development phase of the writing are going to directly tie back to this stage. And then you're also going to appraise the quality of the articles that you retrieved in your search. So the appraisals are going to be weighted much more highly for articles that are randomized control trials. They just have a higher quality of data. So you would certainly want to try to limit yourself in that respect. If you have the opposite problem and there is just not a whole lot of data out there, you may need to expand your search to include older articles, or maybe a systematic literature review that you can discuss that says sort of like over the past 10 years, here's the range of sensitivity and specificity that's been identified for these types of devices. So it can be challenging. It really will depend on your sort of how commonly reported your particular test is. But you will be expected to justify the choices for why you selected some articles and not others. You need to make sure that that entire part of that process is very well documented and very systematic, very unbiased, to ensure that your methodology isn't questioned at some point.
Great, thank you, Sarah. Let's go ahead and get to our next question. In case of new device with new features, what are the acceptance criteria?
Yes, this can be incredibly challenging. In some cases, like let's say you have a particular technology that is totally new to the field and there really isn't a direct competitor. In that case, that's where that state-of-the-art section really is helpful for helping you sort of navigate this problem. So let's say that you have a device that is able to make a new diagnosis, like it's a particular cancer biomarker. And there are no other biomarkers out there that use this exact protein, but there are other ways of making that diagnosis. You would want to collect data on those other methodologies to say, well, if I look for the presence of this factor, you know, the range of making a correct diagnosis is anywhere from 95% of the time to 98% of the time. If I use this method for diagnosis, you know, here's the range for that. And then that can be sort of your acceptance criteria. If you don't have someone who is a direct competitor who uses the same biomarker, who uses the same technology, you may rely more heavily on some of those alternatives, but present them each as data that's been extracted independently. So you can say, making this diagnosis through an ELISA-based technology gives you this range. Making this diagnosis through a PCR technology gives you this range. So you might wind up with multiple different ranges for those acceptance criteria. And then in your discussion, in your analysis, that's where you will describe the limitations of the data, that there aren't any new technologies out there, but based on the current technologies, here are the ranges we've identified. And then you'll still be expected to say, and here's how our device compares to all of these other alternatives. So you can also refer to the guidelines to get sort of what is the acceptable cutoff range in order to justify that your technology meets the expected cutoffs to be considered a clinically relevant diagnostic tool. But you are going to have a little bit, you're going to spend a lot more time on justifying sort of that scientific validity and also defining your analytical performance if there really are not any direct competitors for you out there.
So justify.
Yes.
And not in a way, because we've seen in some potential clients or past clients of ours, or where they've just said, because we think it's the best, or because of this, the justification has to be very scientific. The notified bodies are looking for strong, scientific-based, robust justifications, not because we see sometimes a lot of, it's more trying to convince than it is an actual justification based in science or facts or figures. There's more of sometimes the justifications that clients do is more like trying to be persuasive than it is to like actually use facts to justify why they're taking one approach versus another. So look at your writing and look at your justifications with fresh eyes, and just say, if I were the notified body and I read this argument justification for why we're doing it one way or why something's missing, would I accept that? Is it sound? Mm-hmm. So we do see a lot of folks, teams that are trying to justify, you know, kind of based on their own echo chamber a little bit. It's like, because we think it's the best, and don't you? And, you know, the notified body has no skin in the game. They just need a good scientific justification.
And it comes down to sort of making sure that your risk-benefit analyses and your state-of-the-art section, where you define potential limitations of your other alternative testing methodologies, if those are in alignment, that can really make a solid case for why there's a need for your particular device in this entire landscape. If you can clearly define this meets this, there's an unmet need in the field, that this allows you to, maybe it doesn't require refrigeration steps, so that you can do this in the field, or you can do this in resource poor areas. Maybe it's something that is just far more sensitive, so it can detect the particular analyte earlier in the clinical course. So you're going to want to show where your device sits within that landscape, but also kind of try to point out the need for your device. So why do we need a new technology within this landscape? What is the unmet need within the diagnostic field?
All right, we have just a few more minutes here. So if you haven't sent us your questions yet, be sure to do so using the Q&A button. Here's our next question. What if the subject device does not meet one or more safety and performance objectives or acceptance criteria?
This can happen, and we have had this happen with some clients and some devices. So let's say, like using that same example, your particular test is lower sensitivity, but maybe it's detecting an analyte, like compared to your alternatives, that there, I mean, for something like a COVID test, you're going to get a much lower sensitivity from something like an antigen test compared to a PCR-based test. So if you're comparing yourself, if your competitive landscape sort of lumped together antigen and PCR-based tests, that could be challenging, because it's going to sort of automatically skew the field one direction or the other. It may make it harder to meet that bar or to get that to that level of acceptance criteria. But if you, let's say you have direct competitors and you just are lower than they are, you would want to explain why that is and some potential limitations of your particular technology. Maybe it's that the other tests are using an extra extraction step, but yours doesn't use that step. So yes, you're going to sacrifice a little bit in terms of the sensitivity, but it means that you get the results faster. Or it means that you don't need to have an extra piece of equipment in the lab. So it can be done in the field as opposed to sending it off for a laboratory. If you have a point of care test versus a test that requires laboratory analysis, you're not going to have the same sensitivity if it's an at-home versus a laboratory test. But the benefit is in the overall utility of the test. How is it being utilized, and how does that ultimately impact the clinical benefit? So really your goal is to sort of redefine, you want to make sure that you're explaining, yes, I didn't meet that safety or performance objective compared to the competitive landscape, but I still provide a clinical benefit, and here's why.
All right, this is our last question for our session today. This person would like to see some real-world detailed examples.
I can jump in here too first before you address that, Sarah. Is that, you know, we know that in a webinar setting like this, we can't always address your particular issue that you're that you're confronted with within your own organization. That's why we really encourage folks to reach out to us after the webinar, if you have very specific questions that either weren't answered in our webinars or you would like to, do a deeper dive on, because we have the ability to do free consultations with us, with Sarah or me, to work on your particular issues. So we would certainly encourage you to, to reach out to us after the webinar in any way, and Olivia will talk a little bit more about that. And then the other thing that we want to do too is to make sure that you know that if you did ask us questions, we're going to reach out to you after the webinar to see if there's, again, because we want to help you in any way we can. And we've only got so much bandwidth. So, we want to reach out. We want to see what folks are asking out there, because it really helps us. And I know that maybe something that we say or insights we can give might be able to help you as well. So, Sarah, you were going to, I think, maybe try to give a detailed example or some sort of a, something. Did you want to do that?
I think it comes down to sort of the example I'm envisioning is something related to sort of knowing your intended purpose and how that may change your choice for particular performance measures, like specifically related to the CPR. When it comes to the analytical performance report, most tests have pretty similar methodologies. You're going to measure sensitivity and specificity. There may be some certain parameters that are unique to your type of test. So that's pretty standardized. But when you're tying it back to that clinical utility, the intended purpose is really going to help drive your choice of performance indicators. So if you're trying to screen for a disease, that's where you would want to have something like a diagnostic sensitivity and specificity. How often does this test result in a false positive or false negative? If you do have you would want something like a positive predictive value or a negative predictive value. You could even use something like an area under the curve, where you're looking at what percent of patients would be missed by this particular screening methodology, maybe because there is some sort of an interference with the test, or this test doesn't work in children under a certain age, so a lot of tests involving infectious diseases like blood screening, that would come into play. You wouldn't want to make sure that you're choosing something with performance indicators that directly speak to how that would impact the ability to make an accurate diagnosis, as opposed to something like some particular tests actually directly help you identify sort of like that disease monitoring state, where the patient's already been identified, but you're trying to evaluate whether or not their disease is progressing. And that's going to inform your next steps in terms of the patient. So the performance indicators are pretty much the same for disease monitoring as opposed to disease early diagnosis and screening. You're going to look at sensitivity and specificity, negative predictive value, all of that. Once you get into some of these others where you're actually making therapeutic decisions as a result of the test in terms of like grading or classifications, like maybe you're looking for predicting whether or not you're in kidney failure and you're looking at the levels of creatine. You would want to use things like agreement tables to show that there are, for individuals diagnosed with that particular disease, how does the grading, how does this method of measuring creatine sort of correlate with the grades that are given in a clinical setting? So it really comes down to you're the experts on your test. And so by knowing and having a very clearly defined purpose for your test, you can then go through and identify what are going to be the most relevant clinical performance objectives that we can collect.
All right, thank you, Sarah and Laurie. That is all the time that we have today. I hope we were able to answer most of your questions, but if we weren't, we will follow up with you later today by email. You can also email us for a free consult at criterionedge.com if you're looking to discuss your question or your issue further. Thank you again, Sarah and Laurie, and everyone for joining us. There were so many great questions asked here today. Take care, and I hope to see you at our next webinar.